So sexual reproduction in flowering plants, it is one of the important topic in the state syllabus as well as NCERT syllabus. So now we are going to discuss about important topics in IPE section. So first, let us start with the structure of flower. The structure of flower was, so for the first time, explained by the Camerarius in 1690s. So he discovered the sexual reproduction in flowering plants, and then the process of fertilization was explained by the Strasbourg, and then later Navashin discovered the double fertilization in flowering plants, and that was confirmed by the Gignard in the year of 1899. Okay, now let us start with the topic flower. So it is the structure of flower. So here, so this part is known as the thalamus, and calyx is the outermost whorl, corolla is the second whorl, and the male sex organ, that is androecium is the third, and the innermost is the ovary. Innermost is the ovary, that is gynoecium. So here, first let us start with the life cycle of flowering plants. Then we shall move to the IP section, that is process of fertilization as well as mature egg embryo sac, right? And structure of microsporangium and wall layers. Now let us start with the life cycle of flowering plants. సో ముందు లైఫ్ సైకిల్ ఆఫ్ ఫ్లవరింగ్ ప్లాంట్స్ అర్థం చేసుకున్న తర్వాత దెన్ వి షల్ మూవ్ టు ద స్ట్రక్చర్ ఆఫ్ మేల్ సెక్స్ ఆర్గాన్ యాజ్ వెల్ యాజ్ ఫీమేల్ సెక్స్ ఆర్గాన్ ఇన్ డీటెయిల్. సో హియర్ ద లైఫ్ సైకిల్ ఆఫ్ ఫ్లవరింగ్ ప్లాంట్స్ starts with the sporophytic plant body. Sporophytic plant body. So it is a sporophytic plant body. This is the flower, and here the sporophytic plant body is diploid in condition, and in flower male sex organ is known as the androecium, and the female sex organ is known as the gynoecium. So here, if you look into the structure of androecium, so androecium structure చూసినట్లయితే, సో దీంట్లో మనకు మెయిన్గా టూ పార్ట్స్ ఉంటాయి. సో వన్ ఈజ్ ద ఫిలమెంట్. సో దిస్ పార్ట్ ఈజ్ ద ఫిలమెంట్, అండ్ అనదర్ పార్ట్ ఈజ్ anther. Another part is anther. So together anther and filament, so we call it as stamen. Anther and filament ne stamen అని పిలుస్తాము. Like this stamen is the unit of androecium. సో androecium యొక్క యూనిట్ ఏంటిదంటే stamen. సో gynoecium సంబంధించి, సో the unit of the gynoecium is called as the carpel. The unit of the gynoecium is called as a carpel or pistil. Carpel, or we can call it as pistil. And if we observe the gynoecium structure, we will find mainly three parts. One is the style. This one is a style, and this part is a ovary, and this part is a stigma, right? So here anther ni dissect చేసినట్లయితే, if you dissect the anther, so here usually the anther, most of the flowering plants anther is dithecous anther. So dithecous anther means it contains a tetrasporangia structure. Right? So in that tetrasporangiate structure, sporogenous tissue is present. So that sporogenous tissue contains the microspore mother cells. So here, this is the anther structure. So ఈ anther ni మనం డిసెక్ట్ చేసినట్లయితే, డిసెక్ట్ చేసి మైక్రోస్కోప్ మీద అబ్జర్వ్ చేస్తే, సో ఇట్ లుక్స్ లైక్ this, and it mainly contains four parts, that is epidermis, endothelium, middle wall layers, and tapetum. ఇది కూడా మనకు లాంగ్ ఆన్సర్లో ఉన్న 8 మార్క్స్ క్వశ్చన్, వెరీ ఇంపార్టెంట్ ఫర్ ఐబీ. సో హియర్ it is a tetrasporangiate structure. So all the sporangia are located at the corners, right? So each sporangia contains microspore mother cells. That is, so thousands of microspore mother cells are present. In the sporogenous tissue. So now, we are talking about the life cycle of flowering plant. So flowering plants యొక్క life cycle గురించి మాట్లాడుతున్నాము. This is the sporophytic plant body, and it is a main plant body. It is a main plant body which is diploid in condition. And the flower contains androecium and gynoecium. So here androecium contains microspore mother cell. That is, microspore mother cells are present in the sporogenous tissue. So we can call it as MMC or pollen mother cell. Microspore mother cell or pollen mother cell. So this microspore mother cell undergoes meiosis division, that is also known as reductional division. ఈ మైక్రోస్పోర్ మదర్ సెల్ ఏదైతే ఉన్నదో ఇది reductional division. It undergoes reductional division. Or we can call it as meiosis. We can call it as meiosis. That means it produces 4 haploid microspores. That is, it produces microspore tetrad. Okay, and these microspores are associated each other. ఈ మైక్రోస్పోర్స్ అనేవి ఒకదానికొకటి అసోసియేట్ అయి ఉంటాయి అన్నమాట. సో దే అసోసియేట్ విత్ ద మెటీరియల్ నోన్ యాజ్ కాలోస్. సో కాలోస్ అనే మెటీరియల్ తోటి అసోసియేట్ అయి ఉంటాయి. వెన్ the callose is broken down by the enzyme callase, then the dissociation of microspore tetrads takes place. ఈ మైక్రోస్పోర్ టెట్రాడ్ ఏదైతే ఉన్నదో, సో ఇవన్నీ ఒకదానికొకటి అతుక్కొని ఉంటాయి అన్నమాట, విత్ ద హెల్ప్ ఆఫ్ మెటీరియల్ నోన్ యాజ్ క్యాలోస్. సో క్యాలోస్ ఉండడం వల్ల ఈ మైక్రోస్పోర్ టెట్రాడ్స్ అన్నీ కూడా ఒకదానికొకటి అతుక్కొని ఉంటాయి. ఎప్పుడు ఇవి విడిపోతాయంటే, ఎంజైమ్ నోన్ యాజ్ క్యాలేజ్ అనే ఎంజైమ్ ఏం చేస్తుందంటే, ఇట్ బ్రేక్ డౌన్ ద క్యాలోస్. సో క్యాలేజ్ బ్రేక్ డౌన్ ద క్యాలోస్. దెన్ దీస్ మైక్రోస్పోర్ టెట్రాడ్స్ డిసోసియేట్స్ ఈచ్ అదర్. ఒకదానికొకటి డిసోసియేట్ అయిపోద్ది అనమాట. డిసోసియేట్ అయిపోయి, so they are free now. Microspore tetrads are free. And these are termed as microspores, and all the microspores are haploid in nature. That means microspore mother cell which is diploid in condition, the actual reduction division takes place, and the number of chromosomes are reduced to half. That is, microspore tetrads are formed. And later on, these microspores are developed into mature pollen grain. These microspores are developed into mature pollen grain. And here all the microspores are, all the four microspores are, they are functional. All the microspores are functional in nature. And each microspore or pollen grain, we can call it as pollen grain. each microspore or pollen grain contains one vegetative cell and one generative cell. It is known as the two-celled stage. It is known as the two-celled stage. One is a vegetative cell, another one is a generative cell. So known as the two-celled stage. Most of the flowering plants shed at this two-celled stage. About 60% of the flowering plants they shed at two-celled stage. It is important bit for the NEET point of view, right? So here, when this when this microspore or pollen grain, when it land on the stigma. So stigma is a part of gynoecium. Stigma is part of gynoecium. Okay. Right. So this is the structure of gynoecium. So when this pollen grain land on the stigma, so what happens here? Here, if the stigma accepts the pollen, so this part is known as the stigma. This part is known as the stigma. If the stigma accepts the pollen grain, stigma అనేది pollen grain accept చేస్తే, then this pollen grain gets germinated. That means pollen tube will be formed. A pollen tube will be formed. This pollen tube carries two male gametes. These male gametes are result of mitotic division of generative cell. సో పొలెన్ గ్రెయిన్లో ఉన్నటువంటి జనరేటివ్ సెల్ ఏదైతే ఉందో అది మైటాసిస్ డివిజన్ జరుపుకొని టూ మేల్ గ్యామెట్స్ ఫామ్ అవుతున్నాయి. సో దీస్ టూ మేల్ గ్యామెట్స్ పార్టిసిపేట్స్ ఇన్ ద fertilization process. So that is why double fertilization is the characteristic feature of angiosperms. It is a unique feature. In gymnosperms, only single fertilization takes place, right? So here the pollen grain స్టిగ్మా మీద ల్యాండ్ అవుతుంది. ల్యాండ్ అయినాక పొలెన్ ట్యూబ్ అనేది జర్మినేట్ అవుతుంది. దెన్ దిస్ పొలెన్ ట్యూబ్ గ్రోస్ ఇన్ టు ద ఓవ్యూల్ అండ్ ఇట్ రీచెస్ ద మెచ్యూర్ ఎగ్ ఎంబ్రియో సాక్ అండ్ ఇట్ రీచెస్ ద సినర్జిట్స్. ఓకే, సో ఇది ఈ పొలెన్ ట్యూబ్ అనేది మెచ్యూర్ ఎగ్ ఎంబ్రియో సాక్లోకి వెళ్తుంది. ఇది మెచ్యూర్ ఎగ్ ఎంబ్రియో సాక్లోకి రీచ్ అవుతుంది. ఎట్లా రీచ్ అవుతుంది అంటే, so from the microsporangium, microspore mother cells are formed. From the microspore mother cells, microspore tetras are formed. From these, mature pollen grains are produced. In the same fashion, female sex organ is known as gynoecium. It contains megasporangium. And this megasporangium possesses megaspore mother cell. This megaspore mother cell, which is diploid in condition just like microspore mother cell, it undergoes meiosis division known as reductional division. So megaspore mother cell kuda reductional division jarupukoni 4 haploid megaspores will be formed. So 4 haploid megaspores will be formed, and out of the 4 haploid megaspores, 3 cells will be disintegrated. Only one is the functional megaspore. This functional megaspore is developed into mature egg embryo sac, which contains 3 antipodal cells towards the chelazal end, and a polar nuclei, then two synergids and one egg cell. One egg cell, two synergids. These synergids possess filiform apparatus, that is a special cellular thickenings known as filiform apparatus, right? So this pollen tube reaches the synergids. Pollen tube reaches the synergids. Then the pollen tube gets burst and the male gametes are released into the mature egg embryo sac. Thereby double fertilization takes place. So one male gamete fuses with the egg cell, other male gamete fuses with the central cell. Then it results in the formation of zygote. It results in the formation of zygote. Okay. So male gamete female gamete fuse avadam valla so zygote అనేది ఫామ్ అవుతుంది. సో జైగోట్ నుంచి embryo ఫామ్ అవుతుంది మన. సో హియర్ the female gamete fuses with the male gamete and results in the formation of zygote, which is a diploid in condition. So this zygote later on develops into embryo. From the embryo, a plantlet is formed. From the plantlet, so complete plant is developed. That is a sporophytic plant. So here, up to megaspore mother cell as well as microspore mother cell is termed as sporophytic generation, and after the reduction division and up to the process of fertilization, that is known as the gametophytic generation. Okay, this is the life cycle of flowering plants. Now let us talk about the structure of microsporangium wall layers. It is very important for 8 marks question. Okay, right. So here androecium is a male reproductive organ. Androecium అనేది male reproductive organ and it is represented with the symbol A. A అనే లెటర్తో మనం రిప్రజెంట్ చేస్తాము ఆండ్రోషియంని. సో ఆండ్రోషియంలో మనకు మెయిన్గా 2 పార్ట్స్ ఉంటాయి. ఒకటేమో ఫిలమెంట్, వన్ ఈజ్ అ ఫిలమెంట్ and another one is a anther, together known as a stamen. And if the stamen is sterile, known as staminode. So stamen sterile అయితే దాన్ని staminode అంటాము. So that is not useful in the reproduction process. Staminode is not useful in the fertilization process, right? So here most of the flowering plants are dithecous, and there are some exceptions. In some are monothecous, especially Malvaceae family. So China rose or hibiscus. So in the hibiscus, we'll find monothecous anther. That is bisporangiate structure. Whereas in here most of the flowering plants, here the anther is a tetrasporangiate. This is the structure of tetrasporangiate. All the sporangia are located at corners. This is sporangia. This is another sporangia. Like this, we have 4 sporangia located at 4 corners, right? And these are the wall layers. So epidermis, endothecium, middle layer, tapetum. Right? Tapetum is the innermost layer, and before to that. So here. So we have some important points regarding the flower. So flower structure was given by the Chimerarius in the 1690s for the first time. So flower is a modified shoot. And it develops from floral primordia. And primordia develops first into floral bud, then into a flower. And it is a reproductive part of plant. Flower is a reproductive part of plant. Androecium is a male reproductive part, consists of stamens. Gynoecium is a female reproductive part, consists of carpels, or we can call it as pistil. Carpels అనొచ్చు, లేదంటే pistil అని కూడా పిలవచ్చు అనమాట. Androeciumని stamens అంటాం. Right? So stamen is a male reproductive part, consists of 2 parts, filament and anther. So filament is attached to thalamus or petal. It's transverse section of anther. If you observe this transverse section of anther in microscope, it looks like this. So దీన్ని ఒక యాంథర్ని మనం డిసెక్ట్ చేసినప్పుడు మైక్రోస్కోప్లో అబ్జర్వ్ చేస్తే మనకి ఈ విధంగా కనిపిస్తుంది, దాని యొక్క స్ట్రక్చర్ అనేది ఈ విధంగా కనిపిస్తుంది. సో 4 మైక్రోస్పోరాంజియా, టెట్రాస్పోరాంజియేట్. ఇట్ ఈస్ ఏ బైలోబ్డ్ స్ట్రక్చర్ అండ్ ఇట్ ఈస్ a tetrasporangiate. Okay, so 4 sporangia located at 4 corners and they contains pollen sac and pollen grains are present in this anther. Okay, so here now let us talk about the structure of microsporangium, and here usually microsporangium is a bilobed and dithecous anther. 2 thecas are present. In each theca 2 sporangia are present. Total 4 sporangia. So in a cross section of anther is a tetragonal structure consisting of 4 microsporangia, 2 in each lobes. So later 2 microsporangia of each lobe fuses as a pollen sac. And a microsporangium is circular and surrounded by 4 layers. Those 4 layers are epidermis, endothelium, middle layers and tapetum. Okay, epidermis, endothelium, middle layers and tapetum. Outermost layer protects the pollen and help in dehiscence of anther to release pollen. Now let us talk about this wall layers. Okay, right. So here it has mainly 4 wall layers, that is epidermis, endothelium, middle wall layers and tapetum. Okay, so this is the some part of the sporangia structure, so which I have taken from this transfer section of anther. So this part is represented here. So here, that is the first one is the epidermis, and second one is the endothelium, right? Epidermis, endothelium, middle layers and tapetum. So first layer is the epidermis, second layer is the endothelium and third one is the middle wall layer. Okay, and the innermost is a tapetum. Tapetum is a nutritive tissue. So here epidermis contains some specialized cells known as stomia, and endothelium here the cells expands radially. So cell expansion is radially untadi. So they contain some thick wall is present at the cell corners, and at maturity they lost the water. సో ఎండోథీషన్ ఉన్న సెల్స్ అనేవి మెచ్యూర్ అయినప్పుడు దాంట్లో వాటర్ కంటెంట్ అనేది తగ్గిపోయి దే బికమ్ కాంట్రాక్టైల్ ఆర్ ష్రింక్. ఓకే, దెన్ మిడిల్ లేయర్. సో మిడిల్ వాల్ లేయర్స్ యూజువల్లీ ఇట్ వ్యారీస్ ఫ్రమ్ ప్లాంట్ టు ప్లాంట్. దట్ ఈస్ some may contain 3 wall layers, some may contain 5 wall layers. Okay, so middle wall layers are 3 to 5, and the common function for epidermis, endothelium and middle wall layers is the protection and dehiscence of pollen sac. So ఈ మూడిటికి కామన్ ఫంక్షన్ ఉంటుంది. ఎపిడర్మిస్, ఎండోథీషియం, మిడిల్ వాల్ లేయర్కి కామన్ ఫంక్షన్. ఆ కామన్ ఫంక్షన్ ఏంటంటే దే ఇన్వాల్వ్ ఇన్ ద ప్రొటెక్షన్ అండ్ డెహిసెన్స్ ఆఫ్ pollen sac. Okay, so when the anther is young, anther అనేది యంగ్ ఉన్నప్పుడు it involves in the protection. When the anther become mature, it break the or it dehisces the anther and releases the pollen grains into the surrounding environment, right? And these pollen grains are carried by the various agents like wind or insects or whatever it may be the medium, right? So tapetum cells are, so tapetum is a innermost layer. Tapetum is the innermost layer and it is nutritive tissue. So As earlier I said that microspore mother cell produces microspore tetrad, that is as a result of meiosis. As a result of meiosis, microspore tetrad is formed, and that is associated with the callose. And here tapetum produces the enzyme known as callase. సో ఈ క్యాలేజ్ అనేది మైక్రోస్పోర్ టెట్రాడ్ ఏదైతే ఉన్నదో ఇనీషియల్గా ఫామ్ అయినటువంటి మైక్రోస్పోర్ టెట్రాడ్, ఆ మైక్రోస్పోర్ టెట్రాడ్లో ఉన్నటువంటి క్యాలోస్ని బ్రేక్ చేస్తుంది. బ్రేక్ చేసి ఇట్ రిలీజెస్ మైక్రోస్పోర్స్. మైక్రోస్పోర్స్ అనేది సపరేట్ అవుతాయి అనమాట. దెన్ దే డెవలప్ ఇంటూ మెచ్యూర్ పోలింగ్ గ్రైన్స్. రైట్. సో టపేటమ్ ఈజ్ అ వెరీ ఇంపార్టెంట్. సో టపేటమ్ ఈజ్ అ nutritive layer, and it contains so the thick or dense protoplasm is present. Sometimes more than two nuclei are present, right? And the cells are larger. And this is about the microsporangium structure. It is very important for 8 marks question, right? So epidermis, endothecium, middle wall layer, tapetum. So epidermis contains some special cells known as the stomium, and epidermis is the outermost layer and only one cell thick. And below the epidermis, endothecium is present. Cells expand radially, and at maturity they lose the water and become contractile, and radial expansion of cells takes place. And then middle wall layers. So middle wall layers is usually 3 to 5. So middle wall layer, they form 3 to 5 layers. That varies from plant to plant or flower to flower. And innermost is a tapetum. So tapetum is a nutritive tissue, and it nourishes the developing pollen grain. So pollen grains develop అయ్యేటప్పుడు దానికి ఫుడ్ అవసరం ఉంటుంది. ఆ ఫుడ్ టపేటమ్ అనేది ప్రొవైడ్ చేస్తారు అనమాట. రైట్. సో సెల్స్ ఆర్ లార్జ్ అండ్ థిక్. Dense cytoplasm is present, dense protoplasm is present, and more than one nuclei is present, right? This is about the structure of microsporangium. Okay, structure of microsporangium. Yes. So tapetum nourishes the developing pollen grains. When the anther is young, a group of compactly arranged homogeneous cells called sporogenous tissue occupies the సెంటర్ ఆఫ్ ద మైక్రోస్పోరాంజియంలో స్పోరోజినస్ టిష్యూ ఆక్యుపై చేస్తే అక్కడ మనకు మైక్రోస్పోర్ మదర్ సెల్స్ ఉంటాయి. ఆ మైక్రోస్పోర్ మదర్ సెల్స్ అనేవి మయోసిస్ డివిజన్లో పార్టిసిపేట్ చేయడం వల్ల మైక్రోస్పోర్ టెట్రాడ్ ఫామ్ అవుతుంది. లేటర్ ఆన్ దే డెవలప్ ఇంటూ పొలింగ్ గ్రెయిన్స్, మెచ్యూర్ పొలింగ్ గ్రెయిన్స్, రైట్? అండ్ దిస్ ఈస్ ద స్ట్రక్చర్ ఆఫ్ ఏ మెచ్యూర్ పొలింగ్ గ్రెయిన్. సో హియర్ in this image, so vegetative cell and generative cell is not shown, and we will find vacuole as well as nucleoli is present in this pollen grain structure, and it is mainly two layered. The outer layer is known as the exine and inner layer is known as intine. So usually exine contains sporopollenin, and intine is made up of cellulose and pectin. It is very important for NEET point of view. Okay, and here some pores are present. These pores are known as germpores. ఈ జెర్మోపోర్స్ నుంచే పొలెన్ ట్యూబ్ అనేది జెర్మినేట్ అవుతుంది. అండ్ హియర్ స్పోరోపొలినిన్ ఈజ్ ఆబ్సెంట్ వేర్ ద జెర్మోపోర్స్ ఆర్ ఫార్మ్డ్. జెర్మోపోర్స్ అనే రీజన్ దగ్గర స్పోరోపొలినిన్ అనేది ఉండదు. సో హియర్ దిస్ పొలెన్ గ్రెయిన్ కెన్ withstand extreme temperature, strong alkali, etc. due to the presence of that sporopollenin. So these pollen grains can be stored as a fossils. Okay, right. So microsporogenesis, as I said earlier, in the life cycle of flowering plant, that is the process of formation of microspores from pollen mother cell or microspore mother cell through meiosis is called as microsporogenesis. The cells of sporogenous tissue undergoes meiosis, that is nothing but reductional division, to form microspore tetrad arranged in a cluster of 4 cells. As each cell of sporogenous tissue has potential to form tetrad, so each cell is microspore mother cell. Whereas in the gynoecium, megaspore mother cell undergoes meiosis. They form 4 haploid megaspores, and out of the 4 haploid megaspores, 3 cells will be disintegrated. Only one is a functional. Here it is in the microsporogenesis. Each microspore mother cell produces the 4 microspores which are haploid in nature, and all are potential, all are functional in nature. And pollen grains on maturation and dehydration of anther. On maturation and dehydration of anther, the spores dissociate and develop into pollen grains, and pollen grains release the dehiscence of anther. So usually the dehiscence is in mostly or usually longitudinal dehiscence takes place. Longitudinal dehiscence takes place. Okay. So here pollen grain is usually called as a male gametophyte. So this pollen grain is spherical in shape, and these are some of the important points regarding the structure of pollen grain, and it is not important for the IP point of view. You can skip this for IP point of view, and it is important for NEET point of view. Remember that structure of pollen grain, outer layer is made up of, inner layer is made up of walt and germopore. So these things are important for the NEET point of view. Next, right. So as I said that each pollen grain contains vegetative cell and generative cell, that is 2 cell stage. In 60% species pollen grains are shed in 2 cell stage, whereas 40% species shed in 3 cell stage, in which generative cell divides mitotically into 2 male gametes. Okay, that is 3 cell stage. So in the 3 cell stage, it occurs in 40% of flowering plants. 2 cell stage occur in 60% of flowering plants. Right. So this is the mature egg embryo sac. It is also important for IP point of view. IP point of view. So here, now let us start with the um, mature egg embryo sac structure. So life cycle of flowering plant lo microspore mother cell అనేది meiosis division జరుపుకొని, సో meiosis is also known as క్షయకరణ విభజన, తెలుగులో క్షయకరణ విభజన అంటాం. So it undergoes reduction division to form 4 megaspores. Out of the 4 megaspores, 3 megaspores will be disintegrated. 3 cells అనేవి మాయమైపోతాయి, ఓన్లీ ఒకటి ఫంక్షనల్ ఉంటుంది. దట్ ఫంక్షనల్ megaspores is developed into mature egg embryo sac. Now let us start the mature egg embryo sac.